My name is Dalroy Chen. I'll introduce the paper submitted to IMETA. The title is Daily Occupational Exposure in Swine Farm Alters Human Skin Microbiota and Antibiotic Resistome. Firstly, let's discuss the background of the study. Antimicrobial resistance, AMR, is a serious global public health problem that is widespread in people, animals, and the environment. Our previous study found that the swine farm environment reshapes the human gut microbiota and the antibiotic resistome, and it was later reported that ARGs can be transmitted between the farm environment and the human oral cavity, or gut via host bacteria. Human skin microbiota is characterized by higher vitality and variability compared with more stable intestinal or oral microbiota, and microorganisms can be transferred from the environment to human skin. Skin is considered to be one of the important reservoirs of ARGs and ARBs. However, the characteristics and extent of ARGs and ARB transmission between the farming environment and human skin are rarely reported. Our previous study found that long-term occupational exposure in swine farm alters human skin microbiota. To further investigate the mechanism of the effect and to clarify the specific time when this effect occurs, we designed two cohorts, that is, farm workers and students, and collected skin samples of volunteers and environmental samples. By comparing the dynamic changes of skin microbiome and ARGs of volunteers in one day, the effects of daily occupational exposure in swine farm were investigated. Alpha and beta diversity analysis showed that exposure changed the structure and diversity of skin microbiota. The skin microbiota of the control group remained relatively stable, suggesting that exposure to livestock-related environments, which is rich in microbes, can reshape a person's skin microbiome to a large extent. At the species level, the relative abundance of low-abundance species changed significantly, while high-abundance dominant species remained relatively stable. Some significantly different species were found at T1 compared to T1. Source tracker analysis found that dust, not swine feces, was the main driver of changes in the skin microbiome after occupational exposure. Microbial tracing and strain phylogenetic relationship analysis demonstrates the potential for extensive microbial exchange between workers' skin and their surrounding dust environment. Thus, microbial exchange between the skin and the dust starts when workers enter the swine farm, and transient dust microorganisms can remain stable on the worker's skin for 5 to 10 hours until they leave the swine farm at the end of their work, T2. Previous investigation of the effects of swine farm exposures on the human gut microbiome linked ARG transfers with environmentally mediated microbial transmission events. Similarly, skin microbiota shifts in the post-farm exposure skin correlated with increased abundance and diversity in the antibiotic resistome, compared with pre-farm exposure skin resistome. Previous studies have reported some increase in the relative abundance of ARGs in both the human oral cavity and intestine after occupational exposure in swine farm, and our results were similar, particularly in the forehead skin, where the relative abundance of ARGs doubled after 5 hours of exposure and remained stable thereafter. This suggests that exposure can cause enrichment of ARGs in skin, which also suggests that there may be horizontal transfer of ARGs between workers and the farm environment, with the composition of ARGs in skin after exposure trending towards the dust antibiotic resistome. Our source tracker analysis also suggests the environmental dust as the main source of the ARGs, implying ARG acquisition stems from microbiota shifts. The ARG subtypes enriched after occupational exposure belong mainly to the tetracycline, aminoglycoside, and MLSB resistance genes. These ARG types have been reported to be commonly found in animal feces and dust from livestock farms. Notably, we identified the TET-X gene with its variants in ARGs enriched after occupational exposure. Spread of TET-X variants could pose a threat to human health via treatment failure in the future, thus monitoring TET-X and its variants in humans, animals, and the environment is critical. Enrichment of them may be associated with frequent tetracycline use in the studied swine production facility. This implication emphasizes the need for continued improvement of animal husbandry and stewardship practices. We found 25 mobile ARGs with different patterns coexisting in skin and environmental samples in the ARGs enriched after occupational exposure, and found that their genetic environments were similar through analysis, providing evidence for the shared ARGs between human skin and farm environmental microbiota. Combined with the binning results, ARGs can be transferred between the farm environment and worker skin through shared host microorganisms. In this study, it can be concluded that daily occupational exposure in swine farm can change the composition of human skin microbiota and antibiotic resistome. Host microorganisms that carrying ARGs in the swine farm environment can remain stable on human skin, leading to the potential risk of these ARGs spreading to the community.